Hey guys, I hope everything is going well for you. So you just finished your first test. I hope that you guys did well on it and that you've been studying and keeping up with your work. We're moving on to our next unit, which is all about the cell, and pretty much from here on out for the rest of this semester, as far as I can think, we're going to be covering all things cell processes, and just different things that are taking place within the cellular level and below. It won't be until next semester that we start getting into some of the bigger levels of organization, like ecology, populations, things like that. So we need to get a really good, solid background of the cell and what it is and how it's built. And so that's what this particular slideshow is all about. Once again, a lot of this is going to be a review. There's not a lot here that is new from what you learned back in freshman year. So we're just going to have to bring that knowledge back up to the surface. And so just a quick reminder, we've got our levels of organization of life. And so far we've covered the atoms and the molecules and the macromolecules in the last unit. And then this particular unit is going to focus on organelles and cells. And remember that this technical stage, this level of organization, the cell, is where life actually begins. So one of the things that I need you to be familiar with is the cell theory. And so we actually have a lot of different scientists that contributed to what we know about the cell. And it goes back all the way, here we have the 1500s. And I'm sure there's some people that are left off this list. But I do want to point out some people. You don't necessarily have to know the people for test purposes, but some people that you've probably heard of before and that I want you to be familiar with is that we have Robert Hooke over here. And he was actually the first person to use the term cell. He was actually looking at cork under a microscope. We have Leeuwenhoek, and he actually was using a very basic microscope, and he was the first person to kind of look at microorganisms in pond water. I believe he actually called them animalcules, but essentially what he was seeing was living cells looking at some pond water. And then we have these three guys who are actually listed in this information over here. So we have Schleiden, Schwann, and Virchow. And Schleiden was a botanist, a plant guy, and he determined that all plants are composed of cells. Schwann was similar, except for he made the same conclusion about animals and that animals are composed of cells. And then Virchow is the one who's responsible for saying that cells come from other living cells. And so from the work of all of these people and over a long period of time, what we have are these three basic statements of the cell theory. And really just know these, okay? They're very simple. But number one, cells are the basic unit of life. Sometimes you'll see that written as, instead of life, it'll say the basic unit of structure and function. Essentially, the cell is the smallest unit that's alive. The second one is all organisms. Now, the word organism implies a living thing. Remember that cells make up tissues, which make up organs, which make up organ systems, which make up an organism. But also we could have unicellular organisms. But all organisms, or living things, have at least one or more cells that make them up. And then the last one is Virchow's kind of idea that cells come from pre-existing cells. So cells must come from other cells that are living through cell division processes like mitosis and cytokinesis or meiosis. But essentially cells aren't coming from non-living matter, they are coming from other living cells. Now, like I mentioned, the cell is the smallest unit that's able to pretty much be considered alive. It's the smallest unit that can do all of those functions that we actually talked about in Chapter 1. Chapter 1 was all about the characteristics of life, and we looked at things like maintaining homeostasis, responding to their environment, metabolism, and there were several other things. There was a list of like 8 things. And when it comes to the cell, your organisms could be unicellular. That UNI means one, or they could be multicellular. Now, when you have a multicellular organism, whether it be something like an earthworm or a starfish, all the way up to a complex human, when you have multicellular organisms, they are typically, the cells will become specialized to do a certain job. So your unicellular organisms are pretty much kind of your generic, can do it all, whereas your multicellular oftentimes may not do all functions, they just have particular parts and particular enzymes to carry out certain functions. Now cells are tiny. Hopefully we know that most cells, in order to see them, you're going to have to break out a microscope. And so down here we see just a size range. So I don't know how familiar you guys are with the metric system, but this little unit here, a nanometer, is 1/1000th of a meter. And then we get up here to a micrometer. That's what this word is here, okay? You can see that your organelles, some of your bigger organelles, are going to be micrometers, and then your bacteria cells are also measured in micrometers. Your plant and animal cells can be measured in micrometers, but micrometers equals millionths of a meter. So we have billions of meters here with the nanos, and we have millionths of meters here with the micros. And then now some cells can actually be seen with the naked eye, and that's pretty much going to be your egg cells. Those reproductive cells that get fertilized by the sperm. You think about a chicken egg or an ostrich egg, they can actually get quite large. Now, a lot of times just the egg by itself is not necessarily metabolically active. It doesn't really become active until it's fertilized, but typically your cells are going to be very small, and they're going to be tiny enough that we have to use a microscope to see them. And there's a reason that they are small. Okay, cells are small because large cells become very inefficient at maintaining the metabolism that they need to maintain. So as a cell gets bigger, okay, we're looking at two parts to our cell here. We're looking at a surface area to volume ratio. And I know a lot of times your cells aren't cubes, but it's a lot easier to show you with the mathematics of it. But we kind of have two measurements. We have a surface area, which for a cell is essentially the cell membrane. It's the stuff that's on the outside. And hopefully you remember from your math classes that area is measured in a squared unit. And then we have the volume, which is going to be everything on the inside of the cell. It's going to be the cytoplasm and the organelles. And that volume is measured in a cubed unit. And so what happens is when we compare these two, we're essentially comparing the amount of membrane to the amount of cytoplasm and stuff on the inside of the cell. So we have a squared unit and a cubed unit. Now, as the cell grows, both of these are going to go up, but a cubed unit is going to go up faster, okay? And so what happens is, as the cell gets bigger and bigger and bigger, the ratio of membrane to internal contents is going to get smaller and smaller and smaller. So here you have an example, a really small cube here. My surface area to volume ratio is 6, which means there's 6 times the amount of surface area to 1. And then what's happening here is as we're going up, cell gets bigger, ratio gets smaller. Cell gets bigger, ratio gets smaller. And so this is a problem for cells because the bigger the cell is, the more needs it's going to have. It's going to need more materials coming in, like food. It's going to need more materials going out, like waste. Well, as the cell gets bigger and bigger, there's not as much membrane per internal contents. And so what happens is those cells become inefficient. They can't move enough stuff in and out like they need to. And this is why we do not have really, really large cells typically. Like I said, those egg cells normally aren't metabolically active until they're fertilized. And so we must maintain a large surface area to volume ratio. And so this is why a lot of times cells will divide. They grow, they get bigger, but when they reach a certain point where they're not going to be efficient anymore, they will actually divide into two to keep that large surface area to volume ratio. Now, almost the rest of our lesson is going to cover the different types of cells and the parts that we find in our cells. And this may be something that you've never really seen before, but technically, based on our domains of life, we really have 3 different types of cells. We have prokaryotic cells, eukaryotic cells, and then we have those archaeans. Now, there's still a lot of studying going on. You have to understand that these dudes are kind of hard to find. They live in places that are very extreme typically. And when it comes to like your classification, looking at their relationships, they pretty much are kind of like a mix between the two. Now, I do want to point out that structurally archaeans are prokaryotes. So when we're talking about the structures in a prokaryotic cell, your archaeans are going to kind of meet that structure. But if we get into the genetic components of the archaeans, their DNA and RNA similarities often are more closely related to the eukarya. So if you look at this phylogenetic tree over here, a lot of times they're coming off the same branch as the eukaryotes. So while their cell structure is more like the bacteria, a lot of their other sequences and enzymes may be more like the eukarya. So this goes back to also chapter 1. We looked at our 3 domains of life. Remember that domain bacteria has the same kingdom name. Domain archaea has the same kingdom name. These are your common everyday bacteria. These are your extremophiles. Now the key between the two these two is the nucleus. Remember that no, they do not have a nucleus. They lack a nucleus. And you do. They do have a nucleus. And that's really the main characteristic that divides the two. So your bacteria and your archaeans technically would fit under the prokaryotic cells in terms of structure. And then your eukaryotic cells are going to be everything else in domain eukarya, which is your protist, your fungi, your plantae, and your animalia. And so because we don't know a tremendous amount about the archaeans, we're mostly going to focus on these prokaryotes and eukaryotes. Now, as this says, regardless of the type, all cells, and that's a pretty bold statement. I'm not going to say there's 0 exceptions to the rule, but pretty much all cell types, maybe not every cell on the planet, but all cell types are going to contain these 4 structures. Okay, so these are something that you're going to find in a cell, no matter whether it's a bacteria or an archaean or a fungus or a plant. Okay, pretty much all cells are going to have a plasma membrane, which is that outer boundary. We can also call that the cell membrane. As far as I'm concerned, those 2 terms are interchangeable. All of your cells are going to have cytoplasm, which is essentially all the stuff on the inside. It's going to be that kind of gelatinous or jelly-like fluid. It's going to have all different things dissolved in it, like salts and organic molecules like sugars. And then your cytoplasm also suspends your organelles. So there's a fluid called the cytosol that's actually within the cytoplasm, and then you have all the little parts included in that. So your cytoplasm is the liquidy stuff on the inside with all the suspended organelles. All living cells have DNA. Now, the DNA and where you find it may vary, but every living cell has got to have the DNA, which holds the genetic instruction and kind of tells that cell what to do. And then the 4th thing are ribosomes. Ribosomes are little tiny organelles. Now, something that I think is significant and we need to understand is that they're not a membrane structure. They are not surrounded or bound by a membrane, but instead they're small little structures that make proteins for the cell. And you will find those in both bacteria and eukarya. So it's important to recognize that the ribosomes, a lot of times we remember that prokaryotes don't have organelles, and that's not necessarily a true statement. They really don't have the membrane-bound organelles. So ribosomes are something that are common to all cells, even those archaeans. And so now we're going to get into the 2 main types, the prokaryotic versus eukaryotic. Once again, remember that no and you do. And so we're going to take a look at the structures of these different types of cells and then look at the organelles that could be found within them. And so remember that prokaryotes do not have a membrane-bound nucleus, and they do not have other membrane-bound organelles. Like I just mentioned, this is the key word here. They don't have membrane-bound organelles. It doesn't mean they don't have internal parts, because they do. They just don't have the membrane-bound organelles, such as these listed here. Okay, and here in a minute, as we go through the different organelles, we're also going to see what cells they're found in. Okay, so if they have a membrane structure, they're probably not going to be found in a prokaryotic cell. It doesn't mean there's not a membrane. It doesn't mean they can't do the things that the organelles do. For instance, photosynthesis. In a eukaryote, photosynthesis would take place in a chloroplast. That does not mean that bacteria can never do photosynthesis. There are bacteria that can do that reaction. They just don't have a little house for it. A lot of times what they'll do is they'll use little foldings of their membrane. They just don't have a separate little organ or a separate location to perform that function in. So once again, it doesn't mean they can't do the things, they just don't have specialized parts to do them in. Okay, so some other structures that you might find in prokaryotic cells. In bacteria, they have cell walls made of a structure called peptidoglycan. Archaeans may have cell walls, but it's not necessarily this structure. It's going to be unique because of their extreme environments. Some bacteria could have a capsule, which is kind of like a little sticky substance on the outside. A lot of times it helps them kind of get in and try to avoid immune systems. Some bacteria could have flagella, which are little tail-like structures for movement. Now we're going to talk about flagella again here shortly, but I want you to know that the flagella in a prokaryote would be a little bit different than a flagella in a eukaryote, but the same basic function is still there. And then the last thing that you might find, or that we're going to talk about that you might find, are fimbriae, or also known as pili. But these are little short structures that help the bacteria stick, okay? So these are going to look similar to a structure that we're going to talk about at the end of the PowerPoint known as cilia, but these are pili, okay? These are not the same thing as cilia that you would find in a eukaryote. These are a sticking mechanism. They help a bacteria or a prokaryote attach to something else. Now, some other things that you need to be familiar with about a prokaryotic cell is when it comes to their genetic material, I want you to understand that the DNA structure is the same no matter what the organism is. Okay, DNA always has that double-stranded, double helix type of structure. But what's different between the prokaryotes and the eukaryotes when it comes to their DNA is one of them is their chromosomes. Where we have many chromosomes, and you probably, when I say that word, maybe think of an X. Prokaryotes actually only have one chromosome, so all of their genetic material is wrapped up in one chromosome. And what a chromosome is DNA wrapped around proteins. Okay, it's kind of a condensed version. And the shape of their chromosome is circular. So they have a big loop of DNA, and there's only one of them. Now, another big difference is where you find it. So in eukaryotes, remember, they have a nucleus, and so that's where you're going to find the DNA. In the prokaryotes, it's really just kind of hanging out in the cytoplasm, and the area where it's hanging out we call the nucleoid region. Okay, so there's not an actual structure that is containing it, but it is going to be located in a particular area, and we call that the nucleoid. Now, something that prokaryotes can have, but eukaryotes typically don't, is something called plasmids. So all of their genetic instruction for being a living bacteria is going to be in this one circular chromosome up here. But they have other little small pieces of DNA called plasmids. And on these plasmids could be genes that give them some additional traits. And they can actually pass these plasmids from one bacteria to another, which can kind of generate some diversity. But they don't necessarily have to have them to be a living bacteria. Now, remember that both prokaryotes and eukaryotes had ribosomes, but I do want you to know that the structures are a little bit different. They're smaller and built differently in the prokaryotes, but the function is still the same. The ribosomes are still going to be your protein factories. Now, we know that cells in general are relatively diverse, and prokaryotic cells are too. They come in all different shapes and sizes, but they are, as a whole, smaller and simpler than your eukaryotes. They're about 1/10 of the size, and they obviously don't have nearly as many parts as a eukaryotic cell might. But that doesn't mean they're not diverse. In fact, they're actually more diverse in terms of metabolism. You've got prokaryotes that can make their own food. You've got prokaryotes that rely on other things. You've got prokaryotes that can turn like inorganic substances into organic compounds without light. So they're a very diverse group when it comes to how they metabolize and build molecules. They also are very, very diverse in where they can live. You will find prokaryotic cells living in just about any and every environment on Earth. Places that you may find no other life, you might find some of those archaeans. Places like where maybe there's no light, the depths of the ocean, really, really hot, really, really acidic, really, really salty. So things that might kill other cells, you may find some prokaryotes living there. So they're definitely very hardy when it comes to survival, and we find them all over planet Earth. Now, the eukaryotic cells are going to be the opposite. They're going to be larger and more complex because they have more parts. They do have the nucleus. They do have the membrane-bound organelles. And when it comes to the DNA here, once again, we can have chromosomes. Now, we haven't really gotten to cell division yet, but I do want you to know that chromosomes, we only really see them during cell division. Most of the time, our DNA is in the form of chromatin, which is kind of like a lesser condensed form It's what I called the noodle form back in freshman biology. But when we're getting ready to divide, the chromatin super condenses, and what we see are those little X-shaped structures, okay? So where a prokaryotic chromosome, they have one circle. Eukaryotic chromosomes, they have many of these X's, okay? So we definitely have a little bit of a difference here with our DNA. So in a eukaryote, the DNA is found within the nucleus, and the chromosome structure is different. But the DNA itself is still that double helix, okay? The DNA doesn't have a different structure. It's the chromosome that has a different structure. And so in the eukaryotic cells, because they are more complex, we have lots of different parts, okay? So the rest of our lesson today is going to be all about some of the organelles that you can find within cells. Now, some of these could be found in both. Some of these could only be found in eukaryotes or specific types of eukaryotes. And so what I want you to kind of gather as we go through the rest of this is for each part, I want you to know kind of the structure. So what's it made up of, how it's built. And remember, if it's membrane bound, aside from the actual cell membrane, it's probably only going to be in a eukaryote. I want you to be familiar with the function of that organelle. And then I want you to know which type of cells will you find those organelles in, like a prokaryote, eukaryote, plant, things like that. And so each slide is going to be set up to address these, the structure, the function, and what kind of cells contain them. So we're going to start with that cell membrane, and we're actually going to have a whole lesson on the cell membrane and transport coming up next. And so this is just going to be kind of an introduction. But we know that all cells have a cell membrane or plasma membrane. It surrounds the cell, and it's going to be what controls the transport into and out of the cell. So sometimes we call it like the gatekeeper. Now, hopefully you remember from macromolecules that the main basic structure of the membrane is what's called a phospholipid bilayer. Remember those hydrophilic heads and the hydrophobic tails gives us these two layers so that the heads are touching water. But there's also some other components to the cell membrane, like proteins and carbs and other lipids like cholesterol. And so this is a very important cell part because all cells have it, and it's responsible for that transport. Now, some of your cells might have a cell wall outside of that cell membrane, and the purpose of that wall is to provide support and structure. Now, the cell wall is going to vary from organism to organism, depending on which ones have it, in terms of what makes it up. So cell walls are typically composed of polysaccharides and maybe some other components like proteins. In plants, that polysaccharide is cellulose. In fungi, that polysaccharide is chitin. And in bacteria, this is specifically kingdom bacteria, they have the peptidoglycan. So all of these are going to be some carbohydrate structures. Now, I do want you to remember that animals do not have a cell wall, okay? But for those that do, they can vary on structure. Now, something that you'll find in some of these cell walls, especially in multicellular like plants, is they're going to have these little tiny holes called plasmodesmata. And so we want the cell wall to be structural, and it's a rigid structure, but it doesn't mean that your cells are completely separated from each other like in plants. We still want to allow communication, and so there's these little tiny holes or these little tiny channels that pass through the cell wall, and that way they can still pass hormones or ions or molecules in order for cell communication and various functions. All right, the nucleus. Now, of course, the nucleus is only going to be found in eukaryotic cells, and the nucleus is a double membrane structure. It actually has a double membrane nuclear envelope, which is a membrane around it. We have the nucleoplasm, which is going to be kind of like the cytoplasm in a cell. It's just what's in the nucleus. Notice the DNA is in that form of chromatin, typically. We only see the chromosomes when we're getting ready to divide. And so because that DNA is in the nucleus, it's going to regulate the cell activities. Now, there's also another little structure, and a lot of times it can stand alone in terms of an organelle, but it's called the nucleolus. And the nucleolus is a very dense region within the nucleus, and it is actually where ribosomes are assembled. And so this little dense region of the nucleus builds the ribosomes that are going to be out in the rest of the cell. And so we've already kind of mentioned this, but ribosomes are all about proteins. Okay, Okay, ribosomes are the site of protein synthesis. And remember, they are not a membrane structure. The ribosome itself is typically made up of rRNA and protein. It doesn't have a membrane that goes around it. And so we can find these in prokaryotes and eukaryotes. Now, the ribosomes can be in the cytoplasm directly. We call those free ribosomes. Or you could find the ribosomes attached to a big membrane known as the ER. And that ER stands for endoplasmic reticulum. And so we actually have two different types of ER. We have the smooth, and it's smooth because it doesn't have the bumpy ribosomes on it. And then we have the rough, which looks rough because it has the bumpy ribosomes. Now, the rough ER, because it has the ribosomes are making proteins, remember? And so what's happening is these proteins are being made at the ribosomes, and then the ER is this long, wavy network. And kind of think of it like a little transport system. So as those proteins get made, they kind of get moved along this big, wavy membrane network. And as they're getting moved, they may be modified a little bit with chemical reactions that are taking place in the rough ER. Now, the smooth ER has a completely different function because we don't have those bumpy ribosomes, so it's not necessarily dealing with proteins, but instead it actually does some metabolic activity. It can make certain kinds of lipids, it can detoxify, it can store calcium. It really depends on the cell that it's in as to its specific job, but notice it doesn't have the protein function, okay? And because these are big membrane structures, these are going to be eukaryotic organelles. Now, I want to point out something that it's not necessarily in your notes, but in the cell, in a eukaryotic cell, we have what's called the endomembrane system. And the endomembrane system consists of the nuclear envelope, and you see that that's kind of one with the rough ER. It also includes the smooth ER, and that endomembrane system is also going to include the Golgi apparatus as well as vesicles, okay? So the Golgi apparatus is like the UPS. So those proteins and molecules that were being made, they come in on one side of the Golgi, they get packaged, processed, tagged, and they literally get put into these little vesicles and they get shipped to various locations in the cell. And that's what makes them like the UPS, is they're packaging, processing, sorting, and shipping various materials. And they ship them via these little membrane vesicles, okay, which are just little membrane sacs. And so once again, we have a membrane structure, so we're talking about eukaryotes there. All right, another important component in your eukaryotic cells are lysosomes. Now, the prefix lyse, L-Y-S-E, okay, we're going to see this a couple of times. We've actually already seen it once when we saw the chemical reaction hydrolysis or hydrolysis. Lysis means to break or bust. And so essentially what this word means, some means body, so lysosome means break body. These are going to be little organelles that digest. Now inside they have enzymes, okay, and on your paper I included the word hydrolytic because a lot of times when you're looking at the definition or you're looking at a description, it says that they contain hydrolytic enzymes. And that goes back to that process of hydrolysis, which breaks down big molecules, okay? So let's say they have some old strands of DNA or RNA, or they have some old organelles, or just things that the cell needs to do a little cleanup. The lysosomes will bring them into this little membrane sac, and they will break those materials down. Another little membrane structure are your peroxisomes. And I just want you to be aware that in terms of like identification, it's really hard to look at a cell as a whole and identify peroxisomes versus vesicles versus lysosomes because they look very similar. So if you do see cell part labeling, it'll most likely not include the ones that look a whole bunch alike. Okay, it'll be the ones that stand out and have distinct structures. But you definitely need to know the functions, okay? Now, peroxisomes, if you look at the first part of the word, that probably sounds familiar because most people have heard of peroxide or hydrogen peroxide. And what happens is, through the process of metabolism, hydrogen peroxide gets made. There's going to be some activities that are happening inside of the peroxisomes that lead to the production of hydrogen peroxide. Now, hydrogen peroxide is toxic to our cells, and so also in the peroxisome, while it's producing hydrogen peroxide, it also contains an enzyme that breaks down hydrogen peroxide, so that way it doesn't become toxic. So it's just doing some metabolic activity where hydrogen peroxide gets produced, and then they have other enzymes that will break it down. And really, it depends on the specific cell that it is in as to specifically what function it might have, because there can be some slight variation. But it is a membrane structure, and so it is going to be a eukaryotic structure only. All right, up next are vacuoles. Now, vacuoles equal storage. Vacuoles are big membrane sacs, but they're bigger than the vesicles. So lysosomes and peroxisomes and part of the Golgi, we have these little structures called vesicles. They're relatively tiny. Your vacuoles are going to have a larger structure, okay? Now, there are different types of vacuoles. You definitely see a very predominant vacuole in plants known as the central vacuole. It's actually a big part of its structure, and it fills up with water and other substances. In animals, or maybe even in plants and other eukaryotes, you will find some smaller vacuoles that just store food and waste. And then in a very specialized type of vacuole, you will find in some protist type organisms. They're usually freshwater protists. They live in water, and so their little bodies take on water. And what they do is they store that water in these little vacuoles, and then they contract to pump that water out. But regardless of what type of vacuole it is, it's going to be a eukaryotic structure. And then we have a couple of energy-producing organelles. We have the mighty mitochondria. Normally, if anybody learned anything in biology, they know that the mitochondria is the powerhouse of the cell. And the mitochondria is a really unique little organelle. It's a double membrane structure, kind of like the nucleus. It has an inner and outer membrane, and that inner membrane folds to form these little cristae. And then there's a liquidy portion known as the matrix. And one of the unique things about mitochondria is they have their own DNA. They also have their own ribosomes. So they're very, very complex little organelles, and they are the site of cellular respiration. You do need to know more than just powerhouse at this stage. Cellular respiration is the process that produces ATP. It's where we take glucose and we break it down to form energy for our cells. Now, another special organelle is the chloroplast. Once again, it's a double membrane. It also has its own DNA and ribosomes, and the chloroplast also does some energy-producing reactions. In this case, it's photosynthesis. So this is where the light is used to make those organic compounds that we then break in the mitochondria. And so it's got some specialized structures as well. We have these little individual pancakes known as thylakoids. And inside these thylakoids is the chlorophyll. That's that pigment that makes them green. And that's going to be your light-absorbing pigments for photosynthesis. They come in stacks called grana. And then they're also surrounded by a liquidy fluid known as the stroma. And so you will find these in eukaryotes, but you won't find them in all. Like animals do not have chloroplasts because we are heterotrophic. So if it's a photosynthetic eukaryote, like plants and some algae, then they are going to contain chloroplast. All right, a few more structures to go, and all of these structures that remain are going to be microtubule type structures. We have a cytoskeleton, which if you're familiar with what your skeleton does for you, it's going to be about support and help giving you shape. It's pretty much going to be a bunch of protein filaments. Now, we don't have to know the specific names of them, but just know that it's a bunch of proteins that are kind of embedded all throughout that cytoplasm. You can find these proteins in both prokaryotes and eukaryotes, but it may vary from cell to cell. We also have something called a centrosome. Now, this one can be a little bit tricky, and when we get to cell division, we'll talk about it more. But a centrosome is an organizing center for microtubules. And when we go through the process of cell division, I know for sure plant and animal. I don't know for sure when it comes to like fungi. But in plants and animals, they both have a centrosome. And this is where the spindle is produced in cell division. And the spindle are the proteins that are going to attach to the chromosomes and make the cell elongate. Now, in animals, and I don't know why, I've never understood why, but in animal cells, inside of the centrosome are these little microtubule structures called centrioles. So both plants and animals have a centrosome, but only the animals have the centrioles inside of it. But once again, like the centrosome itself, it's going to help produce those spindle fibers. Okay. And then the last part that we're going to talk about are flagella and cilia. Now, earlier we mentioned flagella for bacteria. So flagella is a structure that could be found both in prokaryotes and eukaryotes. And then cilia are hair-like. These are the ones earlier that I said they kind of look like pili, but they are not the same thing. A prokaryote has pili. A eukaryote might have cilia. Pili were for sticking. Cilia are for movement. So they're not the same things. They're not the same structures. But both flagella, which are tail-like, and cilia, which are hair-like, they both are for locomotion. The purpose of both of these is to help a cell move around, like a sperm cell here, or you could have a euglena, you could have a paramecium. The flagella kind of whip back and forth, the cilia kind of beat back and forth, but both of them are going to help organisms or eukaryotic cells move around. And that is going to wrap up our first lesson on cells. I hope that you remember some of that, and so you may not have to study for quite as long if that comes back pretty quickly. But if not, we're going to be spending a couple of days practicing this material in lab. I will be around soon, hopefully, and hope to see you then. And if you have any questions or concerns, just feel free to contact me. Hope you guys have a great one. Bye.